Hello, today we're going to be talking about Meiosis, chapter 11.3. Questions we have for y'all today are: what are the steps for Meiosis 1? What begins to shorten during anaphase 1? What breaks down after the crossing over happens? What is it called when chromosomes don't move and separate? How many cells are the end result of Meiosis 2? Prophase 1 sets the stage for the reductive decision. Mitotic cells have an interphase period that is similar to mitosis G1 as G2 phases. Once interphase is done, germline cells enter meiosis 1. During prophase 1, the DNA coils tighten. The individual chromosomes under the microscope become visible as a matrix of fine threads. Each thread consists of two sister chromatin join at the centromere. Homologous chromosomes become closed in synapses. They exchange segments by crossing over. Synapses. Synapses is the pairing of homologous chromosomes during prophase 1 of meiosis. The sites of homologous pair of chromosomes are brought close together at both pairs, then aligned side by side in the process of synapses. Synapsid homologs are joined along their entire length. Sister chromatids of each homolog are also joined together by cohesion proteins, which are called sister chromatid cohesions. Crossing over. Recombination nobles is a structure that appears at the same time that recombination occurs. Recombination nobles contain the machinery that is necessary to break and rejoin chromatids. Crossing over is when DNA segments are broken and then rejoined at the same site. While also exchanging materials between the homologous, each chromosome arm usually has one or a few crossovers per meiosis. After the crossing over happens, the cytosol complex breaks. Chromatids are held together by cohesion proteins. During the crossover, the sister chromatids remain connected together. At metaphase 1, paired homologous chromosomes are aligned. The end of prophase 1, each homologous pair is locked together at bioviolet. They capture spinal fibers and move to the center of the cell and align at metaphase plates as paired homologous. It occurs because the kinetochores of sister chromatids of each homologous act to capture polar microtubules. From opposite poles, they are attached to homologous but not to the sister chromatids, as occurred at mitosis. This is called monopolar attachment. The kinetochores of sister chromatids produce tension on paired homologous and pulling them to the equator of the cell. That way, each joined paired homologous line up on the metaphase plate.
During anaphase 1, homologs are pulled to opposite poles. During anaphase 1, the spindle fibers begin to shorten. As it shortens, it disconnects the homolog and chiasmata, allowing the homolog to go toward the opposite pole. The release of sister chromatids cohesion is along the chromosomes, not the centromere. This release is the result of the destruction of meiosis-specific cohesion. As a result of this release, homologs are pulled apart, not the sister chromatids. Each homolog moves from one pole to another with each sister chromatid. The genes on different chromosomes assort independently, which results in independent assortment. That happens because a pole may either receive a maternal or paternal homolog from each chromosome pair. And this is an example of the anaphase 1 of mitosis and then anaphase 1 of meiosis. The difference between these two. Telophase 1 completes meiosis 1. At the beginning of telophase 1, the chromosomes have separated into 2 clusters at each pole of the cell. Now the nucleus membrane reforms around each daughter cell. And now each chromosome has 2 sister chromatides attached by a common centromere because the chromosomes within a daughter nucleus had replicated before meiosis 1 began. Cytokinesis may or may not occur after telophase 1. And here's an example of telophase 1 during meiosis 1. Segregation can occur without crossing over. It is a well-known rule that in meiosis 1, homologs are held together by chiasmata and by sister chromatide cohesion. Homologs move to the metaphase plate and move to opposite poles. There is an exception. For example, in fruit fly males, there is no recombination because they are able to do meiosis without chiasmata. This process is called achiasmate segregation. And here's an example. Meiosis 2 is like a mitotic division without DNA replication. For prophase 2 is when clusters of chromosomes enter prophase 2 at the 2 poles of the cells. The nuclear envelope breaks down as the new spindle forms. Metaphase 2 is when spindle fibers form from opposite poles bind to kinetochores of each sister chromatides, which allow each chromosome to migrate to the metaphase plate. This process is the same as the metaphase during mitotic division. Anaphase 2 is when the spindle fibers contract. The sister chromatides are pulled to opposite poles when cohesion complex joining the centromeres of sister chromatides is destroyed. This process is the same as anaphase during a mitotic division. Telophase 2 is when nuclear envelope reforms around the 4 sets of chromosomes. Cytokinesis follows, as the end result of this division is 4 cells. Each contains a complete haploid set of chromosomes, unlike mitosis, which the end result was 2 cells. Errors in meiosis produce aneuploid gametes. Meiosis must be accurate. Failure to do so can cause too many or few chromosomes to be produced. Movement of chromosomes is called disjunctions. Failure to move chromosomes is called non-disjunction. Gamete with improper number of chromosomes is called aneuploid gametes, which causes spontaneous abortion. And here's an example of a normal chromosome looks like disjunction, and this is the non-disjunction, how it doesn't separate, it just stays together. And here are the questions from the beginning. Number 1, what are the steps for meiosis? 1, prophase 1, metaphase 1, anaphase 1, and telophase 1. Number 2, what begins to shorten during anaphase 1? Spindle fibers. Number 3, what breaks down after the crossing over happens? Synaptonemal complex. Number 4, what is it called when chromosomes don't move and separate? Non-disjunction. Number 5, how many cells are the end result of meiosis 2? 4 cells. And a real life example is the formation of sperm and egg cells. That is an example of meiosis. It's when you're reproducing and meiosis makes sure that an egg gets 2 chromosomes from each parent to equal 4.